In today's class, we will study the BMS third phase research methodology subjects 14th chapter collection and presentation of the data. Here in today's class, we will study the only types of data collection, that is primary and secondary. This topic collection and data and representation of data that we are making in two parts. This is the only first part we are studying here, that is data collection only. Okay, now we will study first of all, introduction. In the introduction, the data collection means the gathering of the information is called as a data collection. It is, if you broadly classified, it is mainly two types, that is primary data and secondary data. Primary data means the researcher himself is collecting the information, that is called primary data. Secondary data is nothing but the someone collected that we are utilizing in our research is called as a secondary data. Okay, now in the introduction, the data collection is the systematic process. It is a systematic process of gathering of the information of the research. The quality of research depends on the accuracy and reliability on the data collection. How the data is collected, based on that the research quality, accuracy and reliability is there. And here data collection is broadly classified into primary and secondary data. Now we will study one by one. The first of all, we will study the primary data. The primary data is the data collected by first hand by the researcher directly from the study participants for the specific research purpose. For the specific research purpose, the researcher himself or assistant is collecting the information that is called as a primary data. The method of collection is the observation, interview, questionnaire, clinical examination, laboratory examination and experiments. All these are the methods we are using for the collection of the data. What is observation here? Just simply observing the participants and writing, collecting the information, that is called observation. Interview means one-to-one asking the question, interview, and collecting the information, writing the things, that is the interview. Next is the questionnaire, writing the questions and giving to the participants and tell to fill, that is the questionnaire methods. We are collecting the information. Another clinical examination, as a doctor we are doing clinical examination. After that we are collecting the information, that is called as a clinical examination. Laboratory investigation, through the lab blood or urine samples, whatever the data were collected, that is the laboratory investigation. Experiments, the different animal trials or human trials, that is the experiments. Through that also we are collecting the information. These are comes under the all the primary data collections. Now what are the advantages of here the primary data collection? The first of all, original and reliable, because we are the researcher himself is collecting, that's why this is the original and trustable, reliable data it is there. Next is the specific and accuracy, it is the specific purpose only we are collecting, that is why it is a specific and accurate validity is there. What are the disadvantages? Disadvantage, it is a time consuming, because the researcher himself is going to visit the participant, that's why it may take the time consuming and expensive. It may spend some money because he has to travel, that's why expensive, and require trained persons, because specific questions we are asking, that's why trained person is required. These are the disadvantages. Example is, a researcher collected the blood pressure reading from the hundreds of the hypertensive patient, evaluated the effect of the Ayurvedic medicine. Suppose if you are giving some Ayurvedic medicines, after that you are evaluating how the drug is effective, that is hundred of patients, you are collecting the information. Okay, this is the comes under the primary data collections. This is the example. The next one is the second point is the secondary data collection. Secondary data collection, secondary data are the data already collected by others and used by the researcher for the different purpose. For our specific research purpose, we are utilizing the others collected data, that is called as a secondary data collection. The sources from where we are collecting the data, the first of all, books and journals, different books we are collecting the data and journals, and government hospitals, the specific disease purpose we are collecting the data. In this month how many malaria patient is there, like that we are collecting from the government hospital or the different hospital records, or else the census reports or WHO publications or research article and databases. All these sources we are collecting the data. These are comes under the secondary data collections. Now the advantage, what are the advantages of secondary collections? The first of all, it is a time consuming, because it saves the times as well as the money, because save time and cost. Next one, easily available, because if you will go to the somewhere and collecting the data, no need to travel, no need to spend the time. It is easy available and useful for the literature review and planning of the research. This is about the disadvantages. Now we will study the disadvantages of the this secondary data. It may be outdated because the someone is collected maybe for 1 year, 2 year, 10 years, that we are collecting, that is, it may be outdated data, it may be there, or accuracy cannot be verified because we don't know who is collected, how much that accuracy is there. That may be the problem. Next one is the maybe exactly, not exactly matched, because someone is collected their different purpose and we are utilizing that data. That's why it may be not exactly match. Example is using National Health Survey data, hospital records to study the prevalence of the diabetes. It means the how diabetes is prevalence or how the diabetes spreading, that we are utilizing the data from the National Health Survey or hospital record. That is the example of the secondary data collections. Now we'll study the next point is the difference between primary data and secondary data. That topic we'll study now. Then first of all, the primary data. The primary data is collected by the researcher himself. Secondary data is collected by others. Next is the original data, it is there, and already available data secondary. Next is the more accurate for the study because specific purpose we are using, and secondary data may be less specific. Next is the time consuming, expensive, and secondary data quick and economical. And example is clinical examination. Through that we are collecting data, primary data, and from the hospital record as well as from the WHO we are collecting data, that is comes under the secondary data. Okay, now the applications, where how these data we are utilizing in the Ayurveda as well as the medicines. The primary data, the clinical trials on the Ayurvedic medicines, Ayurvedic Panchakarma studies, whenever we are giving the new trial drugs, that we are collecting the information that is comes under the primary data at the time of clinical trials. The secondary data is the review of classical Ayurvedic literature text. It may be Charak Samhita, Sushruta Samhita, whatever they are using. That may be our published research article, any related research article, or hospital records or national health databases. From there we are collecting the information. That is the application in the Ayurveda. Hope you understood the today's class. If you understood, please like channel and motivate us, and also share to your classmate and subscribe. Thank you.